In this exercise, we will perform searches in the UCSC site on different genomic databases. The UCSC website includes genomes from diverse organisms and is continuously updated. In this session, we will focus on humans, and for example, search for the human interleukin-1 beta protein. UCSC offers its own search tool called BLAST, a version of BLAST, which is used to search for a query sequence against a genome. Open the UCSC Genome Browser link. Click on Tools and choose BLAST. Now choose the human option from the diverse suggested organism. Pay attention to the genome assembly version that you are using. Each genome assembly can differ from the others by the chromosomal location of the genes, the annotation on them, and more. Therefore, it is important to note which version was used. It is not always recommended to use the latest assembly, depends on your needs. For this exercise, we will use the assembly of February 2009, HG19, for the human as it includes more informative data tracks than the HG38 version. Paste the protein sequence to the input window and click on Submit. At the next page, click on the browser. Please note, most of the time you will get more than one result. Therefore, you should pay attention to the output parameters of each result before choosing the one that is most relevant to you. For example, here we will choose the first result as it covers all the 331 amino acids in the query sequence, from amino acid 1 to 331. However, the second result indicates that there is also a close homologue at chromosome 8, which covers the original sequence from amino acid 60 to amino acid 322. Do not forget to also consider other parameters such as percent of identity. Let's go back to our result page. As we just got one result, we will click on its browser link in order to move to the Genome Browser page. The most reliable source to identify genes is the RefSeq database, which can be shown as the NCBI RefSeq track in UCSC. Alternatively, there is a track called UCSC Genes, which is based on other resources including computational prediction of genes. Both UCSC and RefSeq tracks are presented under the Genes and Gene Predictions tracks title. As we will see below, these tracks will not always correspond. Zoom out until you will get the full sequence view in the UCSC Genes track. If you want to zoom in, you can also drag the mouse on the notebook area that you want to keep. The interleukin B is located on the reverse strand as the sign's directions goes from the right to left. Genes that are located on the forward strand have left to right signs. Exons are presented as rectangle blocks, whereas the introns connect them with lines. Untranslated regions in the exons are presented with thin rectangle blocks, whereas the coding sequence are presented with thicker ones. Let's see how many isoforms exist in the UCSC track for this gene. Recall that a gene in higher organisms can produce different isoforms based on alternative splicing of exons. For this gene, the UCSC track shows two isoforms. The first isoform includes 5 exons. The first exon includes the 5' UTR and the last exon includes the 3' UTR, whereas the rest of the exons construct the coding sequence, which is also known as the CDS. The second isoform is identical to the first isoform, except that it includes 2 more exons, giving a total of 7 exons. The first exon and the beginning of the second exon represent the 5' UTR, while the third exon function at this isoform as a CDS. The second isoform gives a longer protein than the first. Note that the BLAST track shows that input sequence covers only the last 3' region of the complete gene. Look at the RefSeq track. Surprisingly, we can see that according to the RefSeq database, there is only a single isoform with 7 exons, meaning that there is no validated alternative splicing event in this gene. Open the conservation track under Comparative Genomics. Evolutionary conservation between different genomes serve as an important tool for defining functional areas in the genome as exon or control areas. This track shows multiple sequence alignment of 30 vertebrate species and measurements of evolutionary conservation. Areas with faster evolution than expected, for example under neutral drift, will be marked with negative brown peaks, whereas conserved areas with slower than expected evolution will be marked with positive blue peaks. You can see that the conservation is a good tool for prediction whether a sequence is exon or intron. We can clearly see that the 7 exons show higher conservation than the introns. Interestingly, if we focus on the 5th intron, we can see that there is a short conserved region. This can suggest that this intron may contain a functional region, for example, a small non-coding RNA. Let's look how clinical information is presented in the browser. Open the Homing Genes track under the Phenotype and Literature title. Click on the green solid block that appears on the browser track to get more information. It turns out that the interleukin-1b can be involved in gastric cancer after H. pylori infection. Click on the Homing identity number. The interleukin-1b Homing page will open. Now we can explore interleukin-1b data in order to understand its relation to gastric cancer. Click on allelic variant that appears on the left menu. We can learn that individuals carrying interleukin-1b 31T alleles are at high risk of hypochloridria and gastric cancer after an H. pylori infection. Let's go back to the browser page. Open the Common SNP 151 version track under the variation title in the full mode. All SNPs are presented with colored lines, with each DB SNP accession number appearing on the left. Coding synonymous SNP, which as you will learn in a later unit, are less likely to be deleterious, is colored green. Coding non-synonymous SNP and splice site SNPs, which may have pathogenic effects, are shown in red. Untranslated SNP has a blue color. Click on the first blue SNP that appears on the left to move to its data page. According to the color scale, in this gene there are no known variations with clinical significance. Click on the dbSNP accession number to open its dbSNP data page. You can see that in this mutation, the C nucleotide is changed to the G nucleotide. Clicking on Frequencies link on the left will show the frequencies of the C and G nucleotides in different genomes. On average, C appears in 80% of the alleles. The UCSC is an endless source of information and will not be able to go over all of its options. However, we hope that this first trial with this genome browser demonstrates its importance, and I encourage you to explore more of its features. Many tutorials about UCSC are available online.